Hi, everyone. Welcome. This is Reza Shadmehr. This is our seminar series on the cerebellum, and it's been 2.5 years since we started this, and I'm so happy that so many of you have contributed to it, and I hope that it's a opportunity for us to help each other out and improve our science together. So today I'm going to be telling you about this idea of potent vectors of the cerebellum, and I'm going to highlight the work of my students, which are shown here in this picture. And I'm in particular going to highlight the work of Amin Fakharian, whose paper I'm going to focus on. It's going to come out in a month or two, and it's going to describe this work in more detail. In addition, I'm going to tell you about Solomon Muller, a postdoc at Columbia University, and my former student Paul Hage, who also contributed to this work. In addition, being at Hopkins, of course, we're surrounded by such fabulous scientists, and this work in particular has really benefited from a number of them, and I want to highlight who they are: David Z, John Krakauer, Sasha Dulac, and I'm particularly lucky to have at my office next to Tim Harris, who is the inventor of NeuroPixels, and then he's been so generous in providing us with technology and tools to use it. Thank you, Tim. Okay, so the main idea that I would like to express for you, describe for you, goes as follows. To understand what a neuron contributes to a behavior, I think it's useful to look at its teacher. Now, we're going to be talking about the cerebellum, and the function of the neurons in the cerebellum is what is our interest. Now, it's useful to begin by what we know about the pathologies that result, the problems that result when there is damage to the cerebellum. This is my former student, Maurice, is holding on to this robotic manipulandum in our lab and is making reaching movements. And you notice that he does a fine job moving to the targets. So if you look at the movements, these reaching movements of a patient with cerebellar degeneration, what you see is that the movement starts out okay, but then the end seems to be something wrong with it. And this is nicely shown in this example of Rich Ivory.
And you can see all this now?
Mm-hmm.
Okay.
Another example comes from this recording from David Z, where we see an individual making saccades. So what you see is that the eye movement here, as is shown in red, the saccade starts out more or less normally, but then it just overshoots and overshoots and so forth. So it looks like cerebellar disease symptoms are something telling us something about the computations in the cerebellum, and that those computations seem to be important for how to end the movements. And of course, this is what Gordon Holmes had described. And our objective is to ask, well, what is it computing that's so essential for ending of movements? I'm going to focus on the cell types that make up the cortical part of the cerebellum. And we're going to look at the input that comes to it from the Mossy fibers. Then this three-layer network, two groups of interneurons called molecular layer interneurons type 2 and type 1, and finally the output neurons of the cerebellar cortex, the Purkinje cells. And our understanding of this anatomy is because of the work of Wade Reger, Boris Barber, Mike Heuser, and others. And they're the ones who have really been able to describe for us this inhibitory interactions between these neurons and the output neurons. And what we want to know is that when you look at the goal-directed movement, what information is this network receiving in its inputs? What are the interneurons doing to that information? And finally, what information is being sent out? Let's see. Okay, so my goal is to describe this problem for you. And in order to do so, our model system is going to be this beautiful animal. This is a marmoset monkey that we've been focusing on in my lab. And we're going to be looking at this part of the cerebellum here, lobule 6 and lobule 7, where most of the work that I'm going to show you is going to come from. Now, let's begin with the problem that we want to understand. So I showed you that the deficits associated with control of movements look to do with something with ending the movements. But when you look at the activity of Purkinje cells, for example, here during a saccade, leftward saccade, rightward saccade, what you see is that, well, the cells have some baseline activity, and then some of them pause in a different way, some of them burst, and in general, what you see is that there's a diversity among the cells. There's nothing obvious about what parameters of the movements are being encoded by individual cells. And more importantly, there's nothing obvious about ending of the movements. They're all active long after the movement is over. And if you look at a monkey making reaching movements, you see something similar. So this is a work that shows a macaque making a reaching movement with his arm to the left, to the right, and you see there's some Purkinje cells that pause their activity, some Purkinje cells that increase their activity, but many of them are active long after the movement is over. So for both for reaching and saccades, it really seems like the Purkinje cells, first of all, they're modulated for all movement directions. Some burst while others pause. Many are modulated long after the movement ends, and it's really hard to guess what is it that they're computing. So there's something interesting happens, however, when we just add the activities that I've been showing you. So don't worry about individual cells. Let's look at them in some form of population. And so here's an example of that. So if you take a saccade, let's say, and you see that there's arbitrarily divide the cells into what you might imagine as being bursters and pausers, but you see that individually these groups are active before the movement and then also long after the movement is over. But there's something interesting happening because if you simply add these spikes together, so you just look at the population, you see that there's this interesting activity in the population that ends before the movement ends. So somehow these activities here are canceling each other, but these activities here are reinforcing each other to provide you with that increase in activity that sort of goes away before the movement is over. And if you look at certain directions of the eye movement, you see this burst pause activity that crosses right around the time that the movement should begin decelerating. So that's interesting. Just adding the spikes together gives you something somehow makes sense, whereas individual neurons don't. And if you look at reaching movements, so here's a work from Abby Person's lab. Here's a mouse making a reach, and this is the period when it's reaching out toward a piece of food. You see that just like saccades, there are some cells that pause their activity, some cells that increase their activity. But if you just simply now add their spikes together, you see that there's this burst pause activity that seems to cross zero right around the time when the hand is supposed to reach the piece of food. So when viewed from the perspective of a population, P cells do make some sense. They appear to be in some kind of competition with each other, actively canceling each other's output at one period, but then reinforcing it at another period. That's kind of interesting. Like, why are these cells active if they're only if you just add their activity, you get nothing. So what is going on regarding this idea of cancellation? So this notion of cancellation is something that was first noted in a very different part of the brain, the motor cortex, by Mark Churchland, Christian Shenoy, and their lab. So what they were interested in is reaching movements, and they were looking at monkeys making reaching movements. And they noticed that, before the movement begins, there's this activity that's increasing in many of these cells. And then during the movement, there's some activity as well. So they were wondering, like, why is it that the activity here produces a movement, but it doesn't produce a movement here? And the idea was that there must be some kind of cancellation that's happening here that prevents these spikes to reach the muscles, whereas here, the activity does reach the muscles and produce a movement. So what they suggested is that let's view a neuron as having downstream effects that eventually reach the motor neurons, which then activate the muscles. And if we have this neuron labeled as I, and these muscles labeled as J, then what we can say is that the strength of this activity from this neuron to these muscles, these motor neurons, we can represent it as this vector. Let's call it W. So for this neuron I, there's this vector WI, and we're going to name that the neuron's potent vector. And so the idea is that when you record, in this case from the motor cortex, from some neurons, there's this vector W associated with each one of them. So this is W26 for neuron 26, and this is W3 for neuron 3. And the idea is that now if you take their firing rates and you just multiply by this vector that is constant in time, what you're going to get is some activity that's going to be zero during this period. They're going to cancel each other, but not during this period when the movement is taking place. So if you now add the activity of these cells multiplied by the potent vector during the planning period when you're not supposed to move, the activities cancel. But during the movement period, the same potent vectors, the same weights produce activity that reaches the motor neuron. So somehow there's this cancellation that would take place. Okay, so the principal problem that we have is that maybe there are ways to find these potent vectors, but how would we find them for our neurons in the cerebellum? So the way the problem is solved in the cerebral cortex is as follows. They measure neural activity, they measure behavior or EMG, and then they map these two variables linearly upon each other to get this potent vector. Now, the problem is that these weights, of course, depend on the neurons that you've recorded. So if you record, let's say, from 100 neurons, you can find the potent vectors for them. But if you now add to it 100 other neurons, that's going to change the potent vectors that you have. And I think in the cerebellum, we're blessed with something that is really powerful, and that's the anatomy. And we can use that anatomy to inform our mathematics. So what do you mean, Reza, by using anatomy? Carl Eckerod, some 30 years ago, began to describe the relationship between the inputs that Purkinje cells receive via their climbing fibers and the outputs that they make to the muscles. And what they suggested is that, for example, in this case, if you look at the input that this Purkinje cell receives, it cares mostly about the part of the paw that is touched. And if you look at the output of those Purkinje cells, they go to muscles that pull the limb away when that part of the paw is touched. So the idea is to then first describe what is this climbing fiber input. That would be some activity that we can see via the complex spikes, and then ask what is this output that is being generated and how it affects behavior. And another thing that can really help us in the cerebellum is that not only we can measure the input, but we know that when this input occurs, it completely suppresses the simple spikes that the Purkinje cells are producing. So what we can use is two ideas. Use the climbing fiber input to describe the input space, the teacher, for the Purkinje cell, and then combine that with spike-triggered averaging to estimate what is the potent vector for those Purkinje cells, the downstream effects that they have on behavior. So rather than using this equation to find where the potent vector is, let's come up with a different plan. So here's our plan. Measure the encoding of sensory motor events in the climbing fiber input and form a candidate potent vector for that P cell. Then use spike-triggered averaging to verify that suppression of that Purkinje cell indeed induces a change in behavior consistent with what you believe to be its potent vector. And then finally, because we're going to have to form populations, we have to demonstrate that superposition holds, which means when two Purkinje cells are simultaneously suppressed, let's check that their downstream effects is the vector sum of its two potent vectors. So first point of our step, compute a candidate potent vector for the climbing fiber input. So in this case, we're going to be concerned about eye movements. We have the monkey sitting in the room, and you're going to present some visual stimuli, and you're going to measure the climbing fiber response to that visual stimuli. So for example, when the target is presented at time 0, you see that there's climbing fiber activity for some direction. There's this increase. For opposite direction, there's this decrease. Before a movement takes place, there's also this climbing fiber activity. And again, it shows this increase and suppression when the movement is in a different direction. So for this Purkinje cell, we can represent the climbing fiber input as this field of activity that has a direction, theta, and it has a magnitude, rho, which means we have now a vector, w, that we can assign that says this is the climbing fiber input for this cell. This is our potent vector candidate for this Purkinje cell. For some other Purkinje cell, we would find a different potent vector as shown here. And so the candidate potent vector for cell I is simply a vector that describes the climbing fiber input encoding, in this case, of the visual motor space. All right, so now we want to verify that this has something to do with the downstream effects of that Purkinje cells when it eventually reaches the muscles. How we're going to do this? Well, every time a climbing fiber input arrives, there's not just a complex spike, but there's a suppression that takes place in that Purkinje cell simple spike. So this provides us with a natural perturbation and understanding of then how does that affect behavior. So if a saccade takes place and in our group of Purkinje cells, there are no complex spikes, there's some pattern of simple spike activity. So these are the simple spikes now. But now if that same movement takes place and there happens to be a complex spike, it's going to suppress them depending on the time when the complex spike occurs. So now you're going to see some suppression of that simple spikes. Our question is, how does that affect behavior? So we had, for every Purkinje cell, this vector that describes a direction, in this case, direction theta. We can look at the movement that took place in velocity space, project it onto this vector theta and a vector perpendicular to it. So this is when there was no suppression. Here's when there is a suppression. So there's a slight deviation of the eye when that single Purkinje cell was suppressed. And you can now project that trajectory onto your vector space, and you see that the eye got pulled in the direction of the potent vector, not 90 degrees away from it. And more interestingly, when two Purkinje cells that have the similar potent vectors are suppressed, what you see is that the eye gets pulled more in the direction of the potent vector. Now, what about the magnitude of this potent vector? Some cells receive climbing fiber input that has a low magnitude. You see, their encoding of the visual motor space is described by a small vector. Those Purkinje cells, when they're suppressed, they hardly pull the eye. There is another group of Purkinje cells that have a much larger tuning, much larger potent vector. They pull the eye more. And finally, even ones that have the largest ones, and you see that they pull the eye even more. So there's a relationship between the direction and magnitude of the suppression effect on the behavior with respect to the climbing fiber input. So what happens when a P cell is suppressed during a movement? The eyes are pulled by an amount specified by the potent vector of that cell. What happens when two P cells are simultaneously suppressed? So what we're now going to look for is groups of Purkinje cells that are simultaneously receiving climbing fiber inputs, but have different potent vectors. Some of them have similar potent vectors. Some of them have a potent vector that's 45 degrees or more with respect to each other. So let's see what happens when these two potent vectors are engaged. If the potent vectors are in the same direction, so if the Purkinje cell 1 has a potent vector W1, Purkinje cell 2 has a potent vector W2, if you simultaneously suppress them, you see that the eye gets pulled by an amount that's the sum of those two potent vectors. If, on the other hand, one has a potent vector W1, the other one has a potent vector W2 that's now is more than 45 degrees away, the summation is again a predictor of what happens to the eye. And similarly, when it's in the opposite direction. So what happens when two P cells are simultaneously suppressed? There is superposition of their potent vectors. Okay, now we're ready to form a population response. So what we have is that for cell I, Purkinje cell, that you made a movement in some direction, you measure the firing rate for that cell, and you have a potent vector for that cell. All right, this is what you believed is the downstream effect. Those spikes that you saw are going to move the eye in some direction. And what you can do is now you can look at the data that you recorded, and you say, I see that there's a whole bunch of Purkinje cells that are recorded that have their potent vectors over here. But you see that, well, the data that you recorded was not a complete measure of the cerebellum, because you see that the data is biased. So you've recorded from some parts of the cerebellum and you've seen some Purkinje cells, but apparently what you recorded from wasn't the entire population. It's very unlikely that the cerebellum is biased, as I've shown you. So what we need to do is make our data unbiased. And how do we do that? What we're going to assume is the following. We're going to imagine that for every Purkinje cell that we recorded, there exist other Purkinje cells that fire in a similar way, but they have a potent vector that's a rotated version of the one that we recorded. So if we recorded this Purkinje cell, we imagine there exist other Purkinje cells that have similar firing rates, but pulled in different directions. This way, we make our data unbiased. So if we record it from cell I, we're going to imagine there exist these sister Purkinje cells that have similar firing rates, but pull so that we have an unbiased sample. So here's this unbiased sample that we generate, and now we simply add the sum activity of our Purkinje cells to get the behavior induced by the population. This gives us the downstream effects that we expect. So here's what the data set looks like. You recorded some Purkinje cells that provided a burst as a movement took place, some that provided a pause, and you notice that their activity long outlasted the movement. Now we use these equations to plot the downstream effects that we have using their potent vectors, and we see something rather interesting. The potent vector is a two-dimensional vector. It has two components, the component that pulls in the direction of the movement and the component that's noise, which is perpendicular to that direction. And what we see is that there is this activity that helps accelerate the movement, and then the activity precisely crosses zero at the time when you're supposed to begin decelerating, aiding in the process of deceleration, and then completely goes away when the movement is over. Most interestingly, there is no contribution when the movement is 90 degrees away from the potent axis is 90 degrees away, which means that despite the fact that the Purkinje cells are active in all directions, the activity that is not going to be helpful in the direction of the movement is getting canceled. Now, we had these bursters and we had these pausers, so we can divide our cells and ask, are they doing something different? And what we find is that no, they are reinforcing each other's. So despite the fact that the pausers never show the burst, if we simply look at the downstream effects via the potent vectors, we see that they are doing exactly the same thing as what the bursters are doing, reinforcing the movement during the acceleration period, stopping the movement during the deceleration period. So there's two kinds of spike cancellation that's taking place. There's cancellation of spikes that occur after the movement is over. These spikes here, you see that they're gone. And second, there's cancellation of spikes that would have pushed the eyes in the wrong direction here. And so it appears that the Purkinje cells are predicting when to stop the movement. So this may seem a little bit odd. So what do you mean by spike cancellation? Where exactly is this spike cancellation taking place? Let me walk you through an anatomical model that produces a conjecture, what I think is happening here. So anytime one is recording from the cerebellum, if one is recording, let's say from the left side of the vermis, there's some neurons, some burst, some pause, that have a potent vector to the right. Some on the right side have a potent vector to the left. What this means is that they project onto the nucleus, in this case the vestigial, which are going to have excitatory neurons that then cross the midline to a group of premotor neurons. These are the burst generators. They constitute an inhibitory and an excitatory group, which then converge onto the motor neurons of the eye. There's a lateral rectus motor neurons and the medial rectus motor neurons in the case of this horizontal movement that I'm going to walk you through. So let's say we're going to ask the monkey to make a rightward saccade. Let's see what happens. You have some bursters and pausers on the right side that have a potent vector, bursters and pausers on the left side that have a potent vector. They produce a burst, some of them produce a pause, and similarly on the other side. If you now look at the sum of their activity when they reach the nucleus, what you see is that there is an increase on the right side that's early, an increase in this case, the inhibition from the Purkinje cells that's late. So it's just simply a timing difference between the two sides of the cerebellum in terms of the nucleus. Now, if we just follow what happens in the next group of neurons, the inhibitory and the excitatory interneurons, before we get to the muscles, we see that for the agonist, this is the right lateral rectus, the effective contribution is an excitatory input that becomes an inhibitory input right at the time when the movement is supposed to decelerate, whereas for the antagonist muscle, the medial rectus, it is an inhibition that becomes an excitation right at the time when you're supposed to decelerate. But the most interesting cells are the ones that are not the ones that I showed you. The most interesting cells are the Purkinje cells that are going to introduce noise into your movement. So we have a group that have a potent vector that says make a vertical saccade upward, and we have a group that says produce a vertical saccade downwards. These Purkinje cells are not silent. They are going to be bursting and pausing for this movement as well. So they're going to have a some of them are going to burst. For them, this movement is minus 90 degrees with their potent vector. For others, the same movement is plus 90 degrees with their potent vector. Some are bursting, some are pausing. They're going to get to the nucleus and they're going to produce some increase in inhibition. But what then happens is that by the time this effect reaches the motor neuron, they're precisely canceled. So what the result is that half of the Purkinje cells, all of these cells here, are active not to induce a movement, but simply to cancel each other's effects on the movement. Okay, let's now shift our attention to these molecular layer interneurons, the ones that are supposed to transform the input from the Mossy fibers to the Purkinje cells. So unlike the Purkinje cells, we don't have the climbing fiber-induced suppression that we could use for spike-triggered averaging. So what can we do? Again, we think that what's important is think about the anatomy. And I want to show you George Augustine's work that describe how we can use take advantage of anatomy in our case. So we know that the molecular layer type 1 neurons are electrically coupled with each other and then connect to the Purkinje cells, but they don't do it everywhere. They do it to specific regions. And so the idea is to use spike interactions as you do simultaneous recordings from these neurons, identify these type 2 and type 1 molecular neurons, at least putatively, use spike interactions to form networks of neurons that are strongly connected to each other, and then ask, what is the computation that's being performed? So here's a recording made by Amin and Alden that describes for you a typical neuropixel summation of the data. So what you see is that there are a group of Purkinje cells here and climbing fiber inputs. And here's their potent vector. Here's another group down here and their potent vector, a third group, and then finally a fourth group. And you see up here, these Purkinje cells have a pretty strong potent vector that pulls to the left. By the time we get down here, the potent vector is petering out and there's nothing down here at all. Now, if we map on to the molecular layer interneurons and the Mossy fibers, we see that there's also, neighborhoods here, that there's these interneurons and so forth. So what we're interested in is asking, are there these cliques that describe strong interactions between these neurons? And I'm going to show you there are. And the way we're going to define cliques is that within a clique, the simple spikes of the Purkinje cells strongly synchronize with each other. In this case, you see there's a 15 hertz synchrony within the clique, but there's almost no synchrony between the Purkinje cells that are neighboring cliques. So despite the fact that these two groups of Purkinje cells are pretty close to each other, they have no synchrony. The ones that are within a clique, however, they have strong synchrony. The same is true between MLI1s. The MLI1s suppress the simple spikes within a clique, but not between cliques. MLI2s suppress MLI1s between cliques, but not within cliques, but not between cliques. And finally, the climbing fiber input produces a spillover to MLI2s within a clique, but not between cliques. So what this suggests is that there are these small networks of neurons that are contributing together to some downstream effect. And what we're going to do is we're going to say we're not going to put everything together. We're going to divide our networks in these small populations. I'm going to ask what they're doing. And so our critical assumption is within a clique, all the cells have the same potent vector. So we now describe neighborhoods. And then we said that the teacher for that neighborhood is the climbing fiber. And we're going to ask, does that help us make sense of these molecular layer interneurons? So here's our network. MLI2s inhibit MLI1s, MLI1s inhibit Purkinje cells are the output. Okay, now here's the puzzle. When you look at the activity of the MLI1s, they're supposed to inhibit the Purkinje cells, right? But look, they're just bursting in all directions. And there's a slight difference in the timing of when the burst begins as a function of direction of the saccades. MLI2s are supposed to be inhibiting MLI1s, but they're also bursting in all directions. And for them, there's also a slight change in the timing of when that burst begins. So the difference here is not about the difference in terms of the firing rates. It's about the timing of when things are beginning. So remember what the Purkinje cells were doing. We described the output by having basically a potent vector that says, begin accelerating, now begin decelerating at this precise time, and cancellation for spikes that are 90 degrees with respect to your movement. And we want to understand, how could this help us produce something like this? So our assumption is that potent vector is going to be inherited by the inner neurons that make up this network. So here's the firing rate that you measured for some molecular layer inner neuron when the movement was made in this direction. And here's the potent vector that you inherited from the Purkinje cell that you're going to project to. We're going to use this to now say, what is the downstream potential effect of those groups of cells? And something remarkable takes place. Using this equation, we transform this activity into an activity that looks like this, that says what happens is that the MLI1s are releasing their inhibition, which coincides with this increase in activity of Purkinje cells, then increasing their inhibition, which coincides with this decrease in them. So you see things make sense again in the time scales and the mechanism of the potent vectors. MLI2s do what you would expect now that we have MLI1s. So MLI2s are increasing their inhibition coincident with the time when MLI1s decrease their activity. So using the potent vector mapping, we see that MLI1s negatively correlate with the P cell output. Okay, this was our network. The final step for us to ask is, how is it telling the rest of the brain when to begin decelerating? It must have information in the Mossy fibers that are associated with what the goal of the movement is. Indeed, when we look at the Mossy fibers, we saw very nice representation of where the target is. Some Mossy fibers are presenting the cerebellum with information regarding the location of the target. Other Mossy fibers are providing the cerebellum with information regarding what are the commands that are being produced to produce this movement. So we get two kinds of information. Where is the goal? What are the commands that I'm producing to generate this movement? And now let's look at the Purkinje cell output as a function of the amplitude of my movement. Where is the goal? If my goal is at 1 degree, you see that the Purkinje cell output crosses 0 at when that 1 degree saccade is supposed to stop. If my movement is at 2 degrees, you see that the activity is larger, the deceleration should be greater, but again, the timing is time locked to when the movement is supposed to decelerate. Here's for 3 degrees, and here's for 5 and more degrees. So what you see is that the Mossy fibers tell the cerebellum where the goal is located, as well as the commands that are being produced to achieve the goal. The P cells provide the information about how to decelerate and how to stop at the goal. So this is what happens when things are working well. Now, what if we prevented the cerebellum from knowing where the target is? Could we do that? There's a natural condition when this happens. So if you look at the Mossy fiber activity, if the target that the monkey is making a saccade to is associated with reward, there's this really beautiful, robust encoding of where that target is. But if the monkey makes that same movement, but now the reward is not associated with this movement, just a spontaneous saccade that they make, you see that the goal information is now muted. On the other hand, if you look at the copy of the motor commands that are being generated, you see that it doesn't matter if the movement was made regarding whether there's a reward or not. As long as you're making the movement, the cerebellum is getting information about the motor commands. However, it is getting muted information if the movement had no reward associated. So if you simply now integrate the number of spikes that you have during the movement, what you see is that as a beautiful predictor of the amplitude of that movement. So when no reward is at stake, the cerebellum receives poor information about the goal location. So it's a wonderful example now that we can use to say, what does the cerebellum tell the rest of the brain if it doesn't know where the goal is? So let's look at the results. So here's when the movement is reward relevant. You see this beautiful crossing at 0, but if the movement is reward irrelevant, the cerebellum doesn't produce this release that you see at time 0. So basically the ability to predict when to stop the movement, if that's the correct interpretation of what this means, is missing when I don't have reward. When goal information is missing, the P cells no longer aid in decelerating the movement. So what does reward do to this circuitry? Knowledge of where the goal is. How does that affect the ability of this circuitry to function? I just showed you that the deceleration command, it isn't there when the movement is not reward relevant. But there's something actually much more interesting about the role of reward in making this system work. So I've been describing to you the firing rates in this 2-dimensional space. the dimension that helps the movement in the direction of the movement. But there's also this 90 degrees away noise, basically, that's being generated by the cerebellum that's going to push you away from the intended movement. So let's see how precisely those spikes get canceled under these two conditions. We have reward relevant saccades that are being made, and we're going to look at Purkinje cells and the interneurons. As the amplitude increases, what we're going to look at is that the component of the potent vector that's going to pull you away from the target, and you see that it gets better, which means spike cancellation gets better as the movement gets bigger if there is reward. On the other hand, if that movement was irrelevant for reward, then what you see is that spike cancellation gets worse. So when there is no reward at stake, spike cancellation degrades and the cerebellum injects noise into the movement. Somehow, presence of reward makes the circuitry so that it's more competitive with itself and more precisely is removing the downstream effects that would push the eye away from the target. Okay, let me summarize what I've shown you. A potent vector is an estimate of the effects a neuron has in behavioral space. The climbing fiber of each P cell, its principal teacher, encodes the sensory-motor space in a specific way. This encoding provides an unbiased estimate of that P cell's potent vector as described via spike-triggered averaging. Simultaneous suppression of two P cells produces summation of their potent vectors. The interneurons and the P cells are not randomly connected. They form statistical cliques in which the potent vectors appear to be shared. During a saccade, the Mossy fibers provide information about the goal of the movement, as well as a copy of the motor commands. And the P cell population predicts how to decelerate that movement. And during a typical movement, much of the P cell activities have no effect on behavior because those effects are being precisely canceled by activities of other P cells. All right, presence of reward makes the cerebellar circuitry work better. It more precisely cancels the unwanted rewards. Okay, so who cares? What does this mean? This is my mom. I'm really lucky to have her live close by. She lives down the street from me, and I like many of you guys, I go visit my mom, and when I do, she asks me, what you've been working on, and I describe what I've been working on, what the students have done, and invariably I come to this question with her is that, so what does this mean? What's the effect that you're showing here? And so I imagine if she was listening to this talk, what might she say? And I tried to answer that questions for her. So I would tell her that, look, the current approach for finding potent vectors in the cerebral cortex is to use neural activity, map it onto some behavior, and find potent vectors. What I think we have found is that there's a different way we can approach this problem in the cerebellum. By understanding the sensory motor space encoded by a neuron's teacher, we have a way to estimate that neuron's potent vector. She would likely respond to this, and she would say, but Reza, at the beginning of this, this lecture, you started by talking about cerebellar disease. You showed us a patient, and you described how there's issues with them. We know that there's movement disorders that affects their behaviors, and there's cognitive disorders as well when there's damage to the cerebellum. So what would this person say about this lecture that you gave? And I've been thinking about that as well. And many of us are making progress, I think, in decoding the activity in the cerebellum. I showed you that it's possible to look at the Mossy fiber input and have a sense of what are the goals that the person has, and in this case, the monkey has, in making the movement, and then have a mechanistic logic of what is happening in these layers by the time we get to the new output of the Purkinje cell. So you might imagine, not too distant future, when we have some disease that affects the Purkinje cells. In this case, many of the diseases of the cerebellum kill the Purkinje cells. There may be some way to build a machine that would record from the Mossy fiber inputs, decode the intention that exists in this particular instant in time, and then provide the output that you think is needed to produce this output. But I think we can even go beyond that, because I think that these cognitive disorders, which are often manifested in processes of planning, impulse control, and so forth, that those can potentially be understood as well. And this is the work that my student Eli and Alana are working on, which is basically the idea that by understanding the computations that the cerebellum is performing during a simple task, in this case, a saccade, we form these things called potent vectors. But what you have is like a dictionary to help understand the computations that the same part of the brain is performing during other kinds of things like planning and thinking. Okay, to finish, let me have an overview of our story. To understand what the Purkinje cells in the cerebellum are contributing to behavior, what we did is that we examined their teachers, in this case, the climbing fibers. The resulting potent vectors described a population that collectively computed when the movement should start decelerating. This computation relied on Mossy fibers that described the goal of the movement, as well as the copy of the commands that were produced to generate that movement. And what we were surprised by is that half the cells were active not to actually do anything, but to simply cancel the unwanted effects that other active neurons would have on behavior. Reward made this cancellation process more precise. Okay, I'm going to end with a very short poem by Leonard Cohen that many of you guys, I'm sure, are familiar with. He's a 20th century Canadian poet. He says, "Forget your perfect offering. There's a crack in everything. That's how the light gets in." Okay, I am happy to answer any questions. I think my student Amin is going to call on you. If you have any questions, just raise your hand and I'm happy to answer it.
Rich Ivory has his hand on. Rich, please go on.
Hey, Reza, thanks a lot. I look forward to working my way through the paper to dig into this. But on the reward side here at the end, if I follow right, are you making a prediction that the kind of initial burst or the agonist equivalent of an eye movement will be the same for the goal-directed and the spontaneous, but really the difference will all come about in the deceleration phase? And is that what you see?
That's what we see. Yeah, the initial part of effectively the agonist part that's helping the movement is present. What's missing is the part that would stop the movement.
Sasha, please.
Reza, that was beautiful. And Amin particularly. So a comment and a question. The comment is, so the cerebellum really seems to be involved in saccades only when they're voluntary. And so this reward modulation of climbing fiber modulation of the Purkinje cells would be, is that right? Would be sort of an explanation for that.
Yeah, the climbing fibers, not when they're voluntary, when they're reward relevant. So of course, all saccades are voluntary, I would say, even the ones that are irrelevant. But the climbing fiber activity is almost at baseline for the movements that are being made when they're not associated with reward. Whereas for reward, you see this really strong modulation.
Great. Yeah, I sort of meant, the fast phases of nystagmus really are saccades, and express saccades also, that is stimulus driven, can't help but make a saccade to it, don't seem to modulate the cerebellum as much. And so the question is, well, gosh, it costs a lot for Purkinje cells to modulate their firing. So what a waste of energy.
Yeah, I know. And this is a puzzle that is not just for us. You find it into of premotor neurons that, the excitatory inhibitory burst generators, when you on the horizontal saccades, they are antagonistic with each other, but for vertical saccades completely cancel each other. So in many aspects of neuroscience, I find that there's this apparently wasteful production of spikes that by the time they get to the motor neurons, precisely cancel. And there are theories that suggest that this is something to do with this excitatory inhibitory balance that's required for the health of the neurons that you're projecting onto But it definitely is a puzzle that I don't know the answer.
Farzana, please go ahead.
Hi, Reza. Hi, Amin. Thank you. Thanks so much. So I guess I missed one thing in your talk, Reza. These are corrective saccades or just saccades?
These are all saccades, regardless of if they are what we call primary saccades or secondary saccades. These are just all saccades that we don't find any difference between, as long as they're reward relevant, it doesn't matter if they were primary or secondary. And the climbing fiber input is also quite similar in these cases.
And then a follow-up on that. So we have also climbing fiber due to a prediction error.
Yeah.
Well, how does that fit into this story?
Yeah. So let me show you, go back to this input here. It really doesn't matter if this saccade was because you made an error and you're correcting it, or that you're just simply making a saccade to the target that appeared. In both cases, there's climbing fiber activity. Now, there may be some difference between the end of this movement, if there was an error, versus when you made that movement and there was no error. So typically, if you make a movement and there's an error, that results in a correction. That correction is associated with complex spikes, which are potentially the teachers that say how to make that original movement better. However, there are complex spikes that are there before the movement starts, even if that movement, of course, is completely voluntary. So it is not the case that complex spikes occur just when there's error. They're far more predominant when you're just making voluntary movements. They occur when there's an error, but they also occur when there is no error, you're just making a movement.
The ones that occur due to error, those are supposed to gradually go down. So then in that case, the potent vector will be gone. So I'm wondering if these have different origins of input.
Yeah.
And so the Purkinje cell knows which one instructs the potent vector.
Right. As you know, Jennifer Raymond's work has shown that the timing matters a lot. And so these complex spikes that are occurring as the movement is being generated have a different temporal relationship to the simple spikes as compared to these complex spikes that might occur if they were in response to an error. So I suspect that timing is going to be a critical factor in how that learning takes place. And we just haven't looked in detail and tried to understand the relationship between error and as you know, in our 2018 paper, David Hertzfeld looked at the relationship between the error that was induced and the change in the complex spikes. And we found that, again, it pushed the eye in the potent direction. But the key point being that they don't just occur when you have an error. You also get them just when you're making a movement.
Thank you. I have one more question.
Yeah, please.
Yeah, okay, thanks. So how about the movements that are not directional, like saccades are directional, right? So is this a general computation for the cerebellum, you think? And if so, then let's say I blink, yeah.
Farzana, maybe I can describe to you another movement. I don't know much about eye blink, but I can tell you about tongue movements. So we've been studying licking and trying to make sense of try to understand what does the cerebellum do for control of tongue movements. And in the case of tongue movements, the principle that we are going after is similar. First, understand the climbing fiber activity with respect to encoding of the sensory motor space of the tongue. So the part of the tongue that gets touched, the direction of the movement that the tongue is making. And then what happens when Purkinje cells, they briefly get suppressed when the climbing fiber input comes? What's the difference between the tongue movement that had that climbing fiber activity and suppression versus one that didn't have that? And the idea is that, if these theories hold any value, they should just go beyond, obviously, the relatively simple sensory motor space of eyes and saccades, and help us understand more complicated movements like reaching and, in this case, tongue movements. But that work is in progress. Okay, well, thank you, everyone. Unless there are any other questions, I'm so glad that we get to spend this little bit of time with each other. I hope you have a wonderful day.
Thank you.